This is the cell cycle by Saran Burhan. Eukaryotic cells have a molecular control system. The cycle is controlled by a cell cycle controlled system. This is a division cycle that is a series of stages which takes place in a cell. It has division and duplication of its DNA to produce two daughter cells. As we see here, 90% of it is in interphase, while 10% of it is in mitosis. It regulates through this control system by different stages with internal controls, or checkpoints. These checkpoints have signals which tell the cell to either keep dividing or stop. There are three main checkpoints in the cell cycle that help continue the cycle throughout the system. The first phase is the G1 phase. If the cell passes the signal, it usually completes the rest of division. If it doesn't pass the signal, it enters a non-dividing stage called G0. This is where the cell will be out of the cycle and will not divide. The G1 phase is where the cell will grow and it is still in interphase. Next, it will go through the S phase. This is where DNA synthesis is from. After is the G2 phase. This phase makes sure all the chromosomes have been accurately replicated without any mistakes or damage. The last phase is the mitosis phase, or the M phase. This is the spindle checkout. This determines if all the sister chromatids are attached to the spindles and ready for separation of the cell. Within each checkpoint, there are internal and external signals that provide stop and go signs. The first internal signal is the mitosis promoting factor. This is a specific type of CDK, which we'll talk about later, that works on the cell itself. It has the main job of checking that the DNA replication did not produce any mistakes and if the cell grew enough in G2. If there are no mistakes present within the cell, then MPF will allow the cell to go to the M phase, will make a copy of itself. This is also produced by the MPF because spindles are properly attached to the chromosomes and ready to separate between the metaphase and the anaphase. Because chemical and physical factors can influence cell division, PDGF, platelet-derived growth factor, is an example of an external signal. A growth factor is a protein released by specific cells that influence other cells to divide. PDGF travels short distances and stimulates other cells in its area to divide faster than it usually would. As we see here, there are two examples of when there is PDGF and when there isn't. The one with the PDGF has an increased rate of cell division, while the one without it has a basic growth. As proteins accumulate in the cell, a critical amount of proteins will tell the cell to move on. These cells are called cyclins. Cyclins are a group of proteins that regulate the cell cycle by forming a complex with kinases. Kinases are protein enzymes that control the cell cycle. Kinases are active only when attached to cyclins. That's why they are called cyclin-dependent kinases. CDK is a chemical that speeds up the process of the cell cycle and is found in all organisms. As said before, MPF is a specific type of CDK that works on the cell itself. In this example, we can see how the cyclin will bind together to initiate the CDK. These two together will attach to the target protein so it could regulate the cell cycle. The target protein becomes active after it is phosphorylated. In the end, the cyclin is destroyed and goes through apoptosis. This is when the cyclin is programmed to commit suicide because more and more cyclin is being produced and regulated throughout the cycle. Throughout this process, there are also some downsides. Cancer results from genetic changes that affect the cell cycle control. When the cell passes the G1 and the S checkpoint, it's committed to either completing the cell cycle, to divide, or undergo apoptosis. The problem may happen if the cell senses some DNA damage. When the problem is found, the cell tries to fix it, and the cell will kill itself, so there won't be any mutations. If this checkpoint doesn't work, the cells within the DNA damage will continue to divide, which leads to further mutations and can progress to cancer. Mutations in the cell cycle intervene with the proper cycle and are found in cancer cells. Cancer cells do not go through density-dependent inhibition or anchorage dependence, and they don't require growth factors to divide. As seen in the picture, normal cells will increase the rate of division with growth factors and not when there isn't. But in cancer cells, the cells will still divide with or without the growth factors. Transformation is when a normal cell becomes a cancer cell. Tumors form when masses of abnormal cells clump. If they are abnormal and they stay in the same lump, it is called a benign. If they spread to other organs, it is called a malignant tumor. And finally, metastasis can occur when the malignant tumor will enter the blood vessels and travel around the whole body, spreading the cancer. As shown in the picture, we can see the tumor spreading around, making the normal cells becoming cancer cells. This means that there had to be some type of mutation in the DNA that did not pass through the checkpoints and spread around obsessively so that the cell would divide over and over and over again. This has been The Cell Cycle by Saron Burhan, and thanks for watching. I hope this helped.